So today I'll be talking about addressing health and healthcare disparities in lung cancer. So just to give you a little bit of background about me and give you some perspective about who I am, I grew up in New Mexico, which is a state motto of the land of enchantment. It is a really culturally diverse state with about 50% of the population identifying as either Hispanic or Latino. And there are 23 federally recognized tribal nations in the state versus here in Tennessee, there are no tribal nations that are federally recognized. And so it's a really rich cultural diversity state. It also has a lot of geographic diversity with White Sands National Park shown in the left. Hopefully you guys can see my cursor. I grew up in Los Alamos, which is considered to be the high desert. And so that's at 7,000 feet above sea level. And just for me, a really beautiful landscape of wide open spaces. And if you've never been, I encourage you to go to the balloon fiesta, which happens every fall. There are hundreds of balloons that are ascend into the air, and it's just this beautiful display of color. I think there's nearly 500 balloons that ascend. And just a fun little tidbit, New Mexico is the only state that has the letters USA on its license plate, because apparently people think New Mexico is part of Mexico rather than the United States. But very much so, New Mexico shaped who I am today. And one of the reasons why I do disparities research, despite it being the land of enchantment, which I fully believe in, agree with, it is a population, a state that has a lot of poverty, higher than the US national poverty. US, it's about 12%, but in New Mexico, about 18% of the population lives in poverty. And as we all know, having access to wealth really brings along better health and access to healthcare. And so there are a number of challenges that populations in New Mexico struggle with. But I've been doing disparities research for over 20 years now. I'm dating myself a little bit here. But I started my graduate work focused on Latino and African American populations. So this is something I really believe in and is shaped in part by my childhood. All right, so just to get everybody on the same page, what do we mean by health equity? Well, Paula Braveman provides a nice definition saying that everyone has a fair and just opportunity to be as healthy as possible. And it's important to keep in mind our terminology and remember that not all health differences are health disparities. So for example, we can have two equal things and apply some external force, and we might then have a health difference. Or we might have a health difference or two things that are equal and apply some inequity, and this results in a health disparity. So for example, let's say we have poor health among the elderly versus young adults. That is an example of a health difference, not a health disparity. And maybe a silly example where we might have a higher rate of injuries among soccer players versus the general population. Again, an example of a health difference, not a health disparity. So it becomes a health disparity when we link that health difference to some sort of disadvantage that's often social, economic, or environmental. And this can occur across several domains: race, ethnicity, gender, sexual identity, age, disability, socioeconomic status, or geographic location. And I just want to make sure to say, because we're talking about racial disparities today, that there really is no scientific basis for race. I'm sure you've all heard this over the last couple of years, but just to reiterate, this is something that has been used to define people for a long time, but it is absolutely not grounded in genetics and really is an arbitrary definition of how we might describe populations. And then I just want to make you all aware, in case you haven't had a chance to see it yet or are not aware, the National Academies released last week a report that provides a conceptual framework for how we might use population descriptors in genetics and genomics. You all, I think we've all been on spring break, so maybe you haven't had a chance to see this yet. But the authors state that it is time for us to reshape how genetic studies are conceptualized, conducted, and interpreted. And they acknowledge that this may also be a framework for how we think about research in other scientific areas as well. But it's quite an extensive document, and I'm still going through it, but just wanted to make you all aware of it. And so I really try very hard to be aware of the descriptors and how I describe and talk about disparities research. I'm sure I can do better, but I work very hard and try to be intent about it, or intentional. So health disparities can occur across the cancer care continuum, all the way from risk assessment on the left to outcomes on the right, where we have clinical status, quality of life, mortality, et cetera. And so we can have disparities across detection, diagnosis, treatment, et cetera. So our goal with studying health disparities is to really identify them and eliminate those health disparities and address the determinants and address those upstream causes. And so this will really require interventions that are tailored to each one of these locations across the cancer care continuum. And so in my own work, we think about lung cancer health disparities, and I've developed a conceptual framework that's adapted from the NIMHD conceptual framework, and where we have multiple layers of determinants of health that can occur across the individual level as well as the community or area level. So we have biological, behavioral, the physical or built environment, sociocultural, or individual health. And so it's all of these domains that then can influence outcomes and result in disparities across the life course. But this is one example of conceptual framework that we use in my lab. So lung cancer is the leading cause of cancer mortality across the world. Over 2.2 million cases occur each year, accounting for 1 in 4 cancer deaths, or 25% of cancer deaths. So it is a significant public health problem that we need to address. We also know that lung cancer incidence varies worldwide, with geographic variation having some of the highest here in the Americas, but also parts of South America, Europe, Asia, et cetera. So a large variation in cancer incidence, lung cancer incidence, that occurs across the world. And yet we know here in the United States that there are racial disparities in lung cancer, with African American and Native Hawaiian populations having among the highest risk. So these are data from a landmark paper published by Chris Hyman and colleagues in the New England Journal in 2006, where they used data from the multi-ethnic cohort for 5 different racial ethnic populations. So African American, Native Hawaiian, white, Japanese American, and Latino. And they looked among both men and women, men on the top row and women in the bottom row, and then looked, holding constant, the amount of cigarette smoking. So on the left column, we have 10 cigarettes per day, or half a pack. And on the right, we have 30 cigarettes per day. And what you can clearly see is that the purple and the blue lines, which represent Native Hawaiian and African American, have the highest risk of lung cancer. And particularly, we see that at the low levels of cigarette smoking, at 10 cigarettes per day. And we see those lines really diverge as populations age, where we have this higher risk in African American and Native Hawaiian. And then when we hold constant and look at 30 cigarettes per day, we see the still some separation of these lines, but not quite as striking. And so it's thought that maybe the system is sort of being overwhelmed by all of this tobacco smoking, but clear illustration of racial disparities in lung cancer. It's also well established that tobacco smoking is the primary risk factor for lung cancer. This really, of course, came about with the famous 1964 Surgeon General report, where it was clearly established the health risks associated with smoking. So about 80 to 90% of lung cancer cases are attributable to tobacco smoking. And there are well-established biologic pathways by which we know how these carcinogens result and the genes that come into play, and that ultimately we have a loss of normal growth control and resulting in lung cancer. So well-established biologic mechanisms. And yet we know that there is this elevated lung cancer risk in Black Americans, but it's inconsistent with the tobacco patterns that we observe. So Black Americans tend to smoke fewer cigarettes per day compared to white Americans. They begin smoking at a later age. They have an earlier age of lung cancer diagnosis, and they also take in higher amounts of nicotine. And so despite this, there is this elevated risk in Black Americans. And there was an updated report from the multi-ethnic cohort, and showing this figure on the left, where they nicely illustrated that this elevated risk is, in particular, is much higher at low levels of tobacco smoking, where populations are smoking up half a pack a day or less, so 10 cigarettes a day or less, or even 11 to 20. And so very clear disparities that are occurring here. And so this is what my group is working to figure out, is what is leading to this inconsistency with the tobacco patterns. It's also well established that there's a genetic contribution to lung cancer. There's a landmark study published by Joan Bailey Wilson in 2004, where she nicely illustrated that a particular region on chromosome 6q23 through 25 had some susceptibility loci for lung cancer. So she gathered 52 families that had 3 or more affected individuals and identified several tumor suppressor genes within this chromosomal region. And so this was really the first sort of definitive evidence that we had that lung cancer is not just due to tobacco smoking. There have also been a number of twin studies conducted where the estimates of heritability have been shown to be around 20%, although it varies whether or not you're looking at, say, European populations or Asian populations. I don't know that there is evidence yet that's been published that at least I'm not aware, looking in African populations, but that absolutely needs to be done. So anyway, a strong evidence for a genetic contribution to lung cancer. So the largest study of lung cancer has been performed in European ancestry populations. So this is a large study that was published in 2017, where the authors gathered about 30,000 lung cancer cases and about 56,000 controls, and identified a number of genetic loci across the genome associated with lung cancer, looking overall, and then with particular histologies, adenocarcinoma and squamous carcinoma. And you can see that the gene regions vary a little depending on what histologic subtype we're looking at. But the largest study to date has been performed in European ancestry populations. And we know that there's genetic diversity that occurs across the globe. The top part of this figure shows the allele frequencies and how they vary across different global populations, very clear allele frequency differences. In the bottom, we see that there's genetic variation that occurs across different global populations, with African populations having the highest genetic diversity. So very strong evidence and well-established diversity across populations. And worldwide, we know our genetic ancestry varies by geography. This is data provided several years ago now, but provides evidence showing that just based on using genotyping, we can cluster individuals across the globe based on their continental ancestry, as shown by the different colors in this bar plot. And that nicely correlates with their geography, shown in the bottom principal components plot, where you can see that the populations nicely map to their geographic location, shown in Europe. And then on the right, we have a gendergram showing the clustering that occurs, and the colors mirror the bar plot, again, showing that populations cluster based on their genetic ancestry. And yet our genetics research does not reflect our global diversity. This is probably a figure you've seen. It's been shown many times in many talks over the last few years. But nicely, this illustrates that European populations far and above are what our genetic studies are performed in. And yet this is really at odds with what our global population distribution is, where we're actually European populations are not the major population in the world. And so it really, our current genetics research does not reflect our global diversity. So the challenge is our genomic resources are lacking in diverse ancestry lung cancer populations. The largest genome-wide association study that has been done to date is from 2016, where just over 5,000 individuals were used to conduct a genetics association study for lung cancer. And only just a few loci were identified as associated with lung cancer. And so it's really a challenge to be able to understand the genetic diversity that occurs in African descent populations that we know have the greatest diversity in the world. So it's really a challenge in my work to be able to try to understand what are the drivers for lung cancer in African Americans. And as I said, this is the largest European study is really in stark contrast to the sample sizes that we see for African Americans with lung cancer. And this same pattern occurs. This is a study that was published last year. It's a cross-ancestry study of lung cancer, where now there are 35,000 lung cancer cases and 34,000 controls, and only 8% of this study was African ancestry individuals. So it really creates this sort of bottleneck to understanding how the disease occurs within African Americans. And so as a result, there are very few lung cancer loci that have been identified in diverse populations. This is a figure from a nice review article led by Chris Amos, published in 2018, where at the time, 45 loci had been identified as associated with lung cancer, and the majority of those were in white populations, with only 2 from that single genome-wide association study that I showed identified in Black Americans. So it's, again, just really points to the issue that we have at hand. So in my lab, we're working to try to address this issue. And so we are working to pull together different populations from around the country, from UCSF, from Karmanos Cancer Institute, National Cancer Institute, here at Vanderbilt. And then because I really believe that it's a multi-level or multidisciplinary approach that we need to take to understand disparities, we also have social scientists, such as my colleague, Dr. Marino Bruce at University of Houston, and a biostatistician colleague of mine, Sunil Rao at University of Miami. Although he just moved recently to Minnesota, so I actually need to update this slide. But really, it's experts that are geneticists, epidemiologists, social scientists, and biostatisticians that are working to try to address this problem. So really understanding, as I said, the mechanisms of lung cancer disparities requires multi-level data. So we're bringing together different types of data sources, from surveys to clinical data to geocoded and genetics. And that will harmonize all these different data sources and have this then hopefully is the largest study to be able to try to understand what's causing disparities with lung cancer. So we are using the NHLBI TopMed resource, which is a sequencing resource with over 60% are ancestrally diverse in non-European populations, with 180,000 participants and over 800 million variants. So a really nice, freely available resource to hopefully be able to boost our studies and provide more robust evidence. So taking, while we're waiting for our genotyping to occur with our lung cancer collaborative study, we've taken the largest lung cancer GWAS that's been conducted to date in African Americans and have imputed that to TOPMed. And we confirmed the prior susceptibility genes and then identified some suggestive regions as well, and such as the one on chromosome 15q25 with one of the nicotinic acetylcholine receptors. So I would say sort of stay tuned, and as maybe I can come back again and present the updated results once we have our large genotype sample size that we're working on now. We also leveraged the GTEx resource, which is an atlas of genetic regulatory effects across diverse human tissues. Right now, the resource stands at about 54 different tissues from about 1,000 donors. So this is a large study funded through the NIH Common Fund, where donors gave different tissues, and the NIH has conducted RNA sequencing and genotyping, and really to be able to try to understand how gene expression works with phenotypes. And so because it's a mediator between our genotypes and our phenotypes, such as lung cancer. And I'm fortunate to be here at Vanderbilt, where some of my colleagues have developed some bioinformatic approaches to be able to study associations between gene expression and phenotypes, such as lung cancer, without having to have individual level data. So just using summary statistics. So for example, you take the 54 tissues from the GTEx resource, and you can develop gene expression models for the different tissues. Then you use a covariance matrix with the genotyping data, and you can build that in different populations. And then using your summary statistics, as we've done with African American GWAS, then you can look at associations between your gene expression and your outcome of interest. So this is what we consider to be estimating genetically regulated gene expression. So this is really a two-stage process, where in the first stage you develop your linear genetic predictor, where you're looking at associations between your gene expression and your genotypes, or your EQTLs. And then in the second stage, you take your summary statistics from your genome-wide association study, and with that, then you can impute your associations with gene expression. So really a great way to be able to look at gene expression without having to go and generate that RNA-seq data yourself. So we're working to do this. Unfortunately, most of the models that have been published for being able to do this are built in European ancestry populations, not unsurprising. So we have about 600 unrelated sequenced African ancestry individuals, where we're working to build these African ancestry SNP covariance matrices and be able to then look at the gene level associations between our predicted gene expression and lung cancer. And then my colleague here at Vanderbilt developed sort of an update to this called JTI, or the Joint Tissue Imputation Framework, which really takes functional genomics information and integrates that into this genetically predicted gene expression information. And so it provides sort of a more robust and improvement over the prior models that I just described. So, for example, this is, again, that African American lung cancer GWAS that I previously shared. And those results are shown in the bottom part of this figure. And in the top, we have our gene expression association results, or our TWAS, our transcriptome-wide association study. The different colors represent different tissues, but you can see that clearly we're identifying different signals using this different approach with gene expression. And these are based on the European ancestry models. But it's also known that these can be ancestry specific. And so we've, like I said, we've built these kind of African ancestry covariance matrices, and we do see that there are some ancestry-specific effects happening here, where here we have at the top the European ancestry covariance, and in the bottom we have the African ancestry covariance matrix. And so clear differences in what associations we're examining. And so right now we still are working to get the ancestry matching of the training and the test populations. We are working to do some RNA-seq and genotyping of about 80 lung tissue samples from African Americans. And at that point, then we'll be able to have ancestry matched genetically predicted gene expression. Because as consistent with all the GWAS data, the GTEx resource really lacks diversity as well. Only about 12% of the samples are African American. And so we don't have huge sample sizes, but we're working our best to try to increase the diversity and have tissue specific that's relevant for our study. All right, so that's a bit on the genetics that we're doing in our lab. I'm going to switch gears here and talk about some other work that we're doing and talk about healthcare disparities. So what do we mean when we talk about a healthcare disparity? So this is when you have two people with equal risk of disease and equal harm to benefit ratio are not managed equitably. So this occurs when you have variation in access to care, use of care, quality of care, or insurance coverage. But it is not explained by a variation in health needs, patient preference, or treatment recommendations. So anyway, so this is how we define a healthcare disparity. So in 2011, the U.S. National Lung Screening Trial published their study demonstrating a 20% relative reduction in lung cancer mortality with low-dose CT. So here, this figure shows chest radiography in the red line, which is the standard at the time, versus low-dose CT in the black line. And they nicely demonstrated that there was this reduction in deaths from lung cancer with low-dose CT. And so as a result, this led to the approval of lung cancer screening. And so lung cancer screening was approved by the USPSTF in 2013 with the following eligibility criteria. So you had to be between the ages of 55 to 80, have a 30 smoking pack year history, and if you're a former smoker, you've quit within the last 15 years. So these are the eligibility and really a landmark change and really an exciting time in the field of lung cancer screening. So the benefit is that with screening, it leads to a stage shift from late stage to early stage diagnosis. So these are data from the Nelson trial, which was conducted in the Netherlands and in Belgium, where you can see here we have screening detected cancers, non-screening detected, and control. And then the different colors represent the stage of diagnosis, with the dark blue representing early stage diagnosis of disease. And so you can clearly see that with screening, we get a large percentage of individuals that have an early stage diagnosis. And this is really when you have much better outcomes and longer term survival, and you can have better treatment options as well. So that's really the goal with screening, is to have this shift towards early diagnosis. The challenge is that the National Lung Screening Trial had low racial diversity. Only about 5% of the population was African American in this big lung screening trial, with overwhelmingly white individuals. And yet we know that smoking behaviors vary by race, ethnicity. There's a large body of evidence to demonstrate that there are differences in how either metabolism or nicotine intake, puff characteristics, et cetera, occur across different populations. And so we decided to look at those eligibility guidelines in African American adult smokers. And this was done by with several of my colleagues and led by one of our trainees at the time, our biostatistician student, Sarah Mercado, and really did some phenomenal work. Sorry for the alarms behind me, the sirens. So we used a study called the Southern Community Cohort, which is a racially diverse cohort led by investigators here at Vanderbilt. It occurs across 12 southern states with about 86,000 participants. And among them, it's about 66% self-identify as Black Americans. And so it's particularly unique because individuals were recruited from community health centers across the Southeast, such as this one here in Orangeburg, South Carolina. And the locations are illustrated here in the map. So as a result, it's a racially diverse, but also a low-income and medically underserved population. And it happens to sit exactly where there's a high-risk geographic region for lung cancer. This is a map showing the incidence of lung cancer in 2017, and on the right is the location of the Southern Community Cohort. So clear overlap in the geography. So in our work, we looked at the ever smokers in the Southern Community Cohort, or the SCCS, and we found that African Americans were diagnosed at an earlier age and smoked fewer cigarettes per day, consistent with the national evidence that we were familiar with. So on average, white Americans have a diagnosis around 64 years of age versus African Americans at 59 years of age, with white Americans smoking about a pack a day and African Americans smoking half a pack a day, or 10 cigarettes per day. So as a result, what we found is there are these racial differences in smoking pack years at lung cancer diagnosis. And really there's this concern then of health equity in lung cancer screening. So this is data from about 1,200 incident lung cancers among those 48,000 ever smokers. And you can clearly see the shift in the smoking pack years, with African Americans shown in the kind of orange color. These are colors that JAMA Oncology picked, not us. So we're sort of restricted to their colors. And then in white populations, this sort of gray color and the overlap in the dark gray, but very clear differences in the median number of pack years for the individuals. And this is the cumulative distribution function showing this dramatic shift in the number of smoking pack years. And so as a result, we found that the lung screening guidelines were under-selecting Black Americans diagnosed with lung cancer. Nearly 70% of individuals diagnosed with lung cancer were not eligible among Black Americans versus 44% among white Americans. And so we dug into that a little bit more to find out kind of what was driving that. We see that more Black Americans were being diagnosed below the eligibility age, so below 55. More Black Americans were smoking less than the 30 pack years, and more white Americans had quit for a longer time period than they were eligible for. And so our solution was, we proposed that the criteria be adjusted to be reduced to age 50 and to 20 pack years. And that would help with addressing some of these disparities. And we proposed that this change happen in African Americans, but to leave the guidelines at what they were for white Americans. So that recommendation is exactly what happened, except that it was applied for all individuals, but exactly what we proposed, where the USPSTF dropped the age to age 50 and dropped the number of smoking pack years to 20. And they acknowledge in the statement that it would be especially helpful to both African Americans and women. And then in March of 2021, those recommendations were implemented. And in fact, our work is cited in the USPSTF lung screening guidelines. So the current eligibility criteria are as follows: age 50 to 80, a 20 pack year history, and having smoked in the last 15 years. So myself and others published a statement along with the American Thoracic Society outlining what the disparities are and strategies to be able to address these. There are a number of disparities that occur in lung cancer screening. And in that document, we outlined that the current screening guidelines actually do not ameliorate the disparities. Those disparities remain. So these are data using from the National Health Interview Survey with the 2013 screening guidelines versus the 2021 across different metrics from percent eligibility, percent preventable deaths, life years gained, or number needed to screen. And you can see that really there's still inequity in the screening guidelines. So part of what we're doing at my lab is to try to identify who should be screened for lung cancer and developing prediction models in diverse individuals. So there are a number of disparities, as I mentioned, that are occurring in lung cancer screening. They're really complex. We have a lot of mistrust and stigma. It's the only major cancer that we screen for based on a behavior. All the other major cancers are based on age or your sex. And so there's a lot of challenges because smoking is part of the eligibility criteria. Access to screening is challenging, distribution of screening centers. There's still a lot of a lack of awareness in terms of availability of screening. So we need to educate providers and patients. Having some health literacy and numeracy is a big challenge with lung cancer screening. It's a cancer that requires a shared decision-making with a visit with your provider. You can't just decide to go and get scanned and go and make an appointment yourself. You have to have a shared decision-making visit with your provider. And then insurance status is a big challenge as well. I'll go into that here in a minute, but multiple challenges in terms of screening disparities. And yet the population that we need to be screening really is not yet being reached. Only less than 6% of eligible adults have been scanned to date. There are nearly 14 million US adults that are eligible, and wide variation across the states about how well we're doing. The access is a challenge as well. There's often the poorest access where the highest mortality occurs. The location of screening centers is not necessarily where we need to be doing the most screening. So there are challenges of place and socioeconomic status that really marginalize vulnerable populations. There's wide variation in insurance. For states that did not have ACA expansion, there is no coverage for Medicaid patients. That is true in Tennessee. It really is quite a challenge. And unfortunately, the populations that tend to be smoking are often of low education or uninsured. And so additional barriers in terms of reaching the population that we need to be able to do screening. Additional challenges are that populations living in poverty are often those that have the highest lung cancer mortality, particularly men. So again, challenges of having access and resources to be screened. Another challenge is, as I mentioned, you need a shared decision-making visit with your provider in order to get scanned. These aids are really inadequate for vulnerable populations. The AMA recommends a 6th grade reading level, but many of these tools are above that reading level. It also requires an understanding of numeracy and risk. And very few of these tools are culturally sensitive, such as with Asian Pacific Islander populations, for example. So another area where we greatly need to do some work. So it's really that we need new strategies to ensure equity for lung cancer screening. There's at this point in time really a strong potential for screening to widen lung cancer disparities. And it really needs to be a multi-level and multidisciplinary approach where we can redefine the eligibility guidelines, develop the appropriate shared decision-making tools. It would be great to have a mobile scanner. That's one thing I would love to be able to do, is bring scanning to the population that needs it. And we need to adjust our health policies where we can provide preventative lung screenings for low-income or uninsured populations. And we need to bring more awareness about the availability of lung cancer screening and help patients navigate through the system with navigators, for example. And in fact, this is recognized by the USPSTF that there are still significant gaps that occur in terms of these inequities with screening for lung cancer. So a lot more work to be done. So just to end by saying that a quote by Mary Claire King, where the most daunting task for us is not tackling new discovery, but rather integrating discovery into meaningful social context. And so with that, I really tried to also bring in community members into my work. I lead a small lung cancer focused community advisory board where we've developed some materials to help raise awareness. And this was a great project that an undergraduate student of mine helped with, and where she knows graphic design. And so we met with the community members and got feedback about what the infographic should look like. And it's worked out well. In fact, she applied for medical school, and this was a large discussion, this topic during her interview, she said, and now she's on her way to Harvard. So she was pleased with not the whole process, but it's also been great for her career as well. And with that, I'd like to thank the team and funding that makes all of this possible.